Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about yeast extract peptone dextrose medium. We also called it YEPD medium. It is actually the fungal culture medium which we specially used for the growth of yeast fungi like Saccharomyces cerevisiae, Candida albicans, Leueromyces lactis and Candida neoformans, etc. Now let's talk about the composition of this media. This media is composed of dextrose ingredient, peptone, agar, distilled water and yeast, and these are the amount of these ingredient which are used. And this is the media. As you can see, mostly the media will be available in this form. Now let's see the principle of this component or the function of each component. First of all, we have yeast extract. Yeast extract mostly provide the vitamin B and all necessary amino acid that are used for the fungal growth. We have peptone which act as a nitrogen, vitamins and minerals, and dextrose. These dextrose, just like in subbroad dextrose medium, so in this case dextrose is also present, but this dextrose is actually providing carbon source that are used for the yeast growth and especially for the Saccharomyces cerevisiae. We have agar that act as a solidifying agent that mostly provide a stable environment through which fungi grow in the form of colony, and we have distilled water that mix all the ingredient. Now how to prepare this media? First of all, we have to take the component, or we take the dehydrated powder 65 gram in 1 liter, and after that we have to boil it to completely dissolve the component or the powder for almost 1 minute. After that we have to sterilize it using autoclave machine for 121 degrees Celsius for 15 minutes at 121 degrees Celsius. After sterilization, the media should be cooled, and approximately at 50 degrees Celsius the media should be poured into sterilized petri dishes. Now what type of fungi we get? So mostly we get Saccharomyces cerevisiae that can produce luxuriant growth, and it is smooth, moist, gelatinous colony. We have Candida albicans and Leueromyces lactis that also produce luxuriant growth. Candida neoformans also produce light grey color colonies. Now these are the growth of Candida neoformans. As you can see, with light grey colonies. We have Saccharomyces cerevisiae which are very small colorless or luxuriant growth of the colonies. These are actually very large and luxuriant growth, but in the picture you can't see the luxuriant growth, but if you see it live, so you will found luxuriant growth. Candida have the same, as you can see, the luxuriant growth, and Leueromyces lactis that also make a luxuriant growth. So that's all about the yeast extract peptone dextrose or YEPD medium. Thank you so much for watching.